thank you to the Simons Foundation for inviting me to give this presentation. I'm super excited and welcome everyone. Thank you for joining. So when you hear that the rates of autism are rising, what does that actually mean? Who's being counted? How are they being counted? And why are the rates changing? So today I'll unpack the latest CDC's ADDM network paper to explain not only prevalence, how it's determined, but also what this report reveals about examining age of first diagnosis, telling us who is being identified in terms of race, ethnicity, SES status. These details give us insight into who is being recognized, who is being missed, and why this matters. I'll also use the CDC ADDM network paper as a kind of like a jumping off point for a much broader conversation where I'll highlight new research at the UCSD Autism Center of Excellence and across the field that's reshaping how we think about autism, its causes, its heterogeneity, and its future. So the MMR paper is published around every 2 years or so, where they describe prevalence, age of first diagnosis, demographic trends, clinical characterizations. But as I said, this kind of opens up the opportunity for bigger conversations like why are the rates rising? What really causes autism? Age of first diagnosis. Why is this such an important thing that everybody's talking about all the time? And have the rates changed across time? Demographic trends. Are there really sex differences? What is the heterogeneity in autism? And people are now talking about should we change dramatically the DSM in terms of how autism is diagnosed. But let's start from the beginning. So what is the CDC's ADDM network? The ADDM network stands for the Autism and Developmental Disabilities Monitoring Network. And I'm just going to minimize so I can see everything. And it actually began not that long ago in 2000. It's based on the Children's Health Act, where Congress signed into federal law. It authorized the Centers for Disease Control to create the ADDM network to track the number and characteristics of children with ASD. Its mission is to estimate the number of children with ASD living in different areas of the United States using record review from multiple sources. We track 8-year-olds and 4-year-olds, although today I'm just going to focus on the 8-year-olds so it's not like too redundant and too much. But the methods are very similar. So we describe the population of children living with ASD, including the age of first diagnosis, compare how common autism is across different areas of the country. So we can kind of see if there's a lot of similarities and differences, identify changes in occurrence over time in specific regions, and understand the impact of ASD-related conditions in US communities. So who participates? How is this actually done? Right now, we have 16 ADDM sites, and they're roughly categorized into kind of the West, the Middle, and the East. And what you see on this map, something I didn't quite realize until I joined the ADDM network, is these green areas actually shows you the tracking area, the surveillance region. And you can see that actually it's not the whole state in any of these 16 regions. In some places like Arkansas, it's actually a large portion. It's multiple counties across the state. And in other places like Indiana, it's a very tiny region. And this is because we're actually tracking every child in those geographic regions. Right now, we're tracking 250,000 children across those 16 states. And here on the right side of the screen, you can see that the demographic characteristics of the individuals in this tracking area is fairly similar to the US profile. Whites are a little bit underrepresented, blacks are slightly overrepresented, but it's a nice representation of America. And the way the CDC does it is that we report things. So when you read the paper, it's coming out, let's say in 2025, but they're always talking about the surveillance year, the birth year of the 8-year-old, and then the publication year. So there's 3 kind of dates you have to keep in mind. So here in this example, the most recent paper is based on surveillance year 2022. That is the year that the ADDM network is determining the prevalence for. Then, okay, if we're talking about 8-year-olds in 2022, that means they were born in 2014. So that is the birth year of the kids that are being surveilled. And then it takes around, 2-ish years by the time we're finished crunching the data and analyzing everything and writing the paper before it's published. So this is kind of like the time sequence when you're reading these papers. And so how does it actually happen? How do we really do it? There are 5 critical steps. The first one is we identify places that serve children with autism, like schools, hospitals, treatment agencies, and we try to form these great relationships with every institution in the geographical region that helps and serves children with autism. And then members of the epidemiology team collect records from these institutions regarding who may have autism. And then we check to see, do the children in the records that we receive meet diagnostic criteria or meet a case criteria for autism. And there are only one of three ways that a child can be counted as, yes, you have autism. The first way is, do they have a diagnostic statement from a professional evaluation in their record, usually like, a licensed clinical psychologist, for example. Two is, do they have an autism eligibility in their school record? Or three, is there somewhere in their record an ICD code of autism, like, for example, 840? That's it. Those are the only three ways that you can end up being counted as ASD within this prevalence report. Then identifying information is stripped from the data set, and scientists analyze these de-identified data sets to determine prevalence, age of first diagnosis, and all these other great things that we like to discuss. And then these findings are returned to the community to improve the care of children with autism, like I'm doing right now. I'm going to be going over all of the results. And so these 5 steps are pretty much the basic process. And here's the MMWR. You probably have seen the paper came out recently on April 17th, 2025, led by Kelly Shaw. So first, let's talk about prevalence. Everyone probably knows by now that our rates are 1 in 31. So how is that determined exactly? So basically, we count up all the cases that were identified with autism in the defined geographic region for a given surveillance year. And then we just divide that by all of the total 8-year-old children living in that specific geographic area as determined by census data for that same given year. And then that's how you get the prevalence. But something I really want to point out that people maybe don't quite realize is that this is a population-based approach, which can provide high accuracy because you're literally circling in a particular region, counting every child in that region and determining the prevalence. So it's very expensive, but it's, I think, pretty accurate way to do it. And think about contrasting that with other ways you might want to do it. Like, for example, a national survey. Somebody might mail you a survey. Oh, does your child have autism? Yes or no? Or they might call you on the phone for an interview. But you can imagine that those ways of doing it, while maybe you can canvass largest areas, I think that they're really prone and vulnerable to bias in terms of who wants to answer your questionnaire and who wants to answer the phone. Same thing with autism registries. It depends on like who's going to register. So I think that this population-based approach is a really nice, rigorous way of doing this. So here's kind of a results in terms of the heterogeneity and prevalence across the United States. Here you can see all these different numbers, and what do you notice? There are really big differences depending on the state the child lives in. Here we are, California, we have the highest prevalence rates, a little bit higher than 5%. And I'll talk about why that potentially is. And in comparison to other places like Laredo, Texas, where it's 1 in 103. But when you hear that number 1 in 31, it's an average of all of these, and that's how we get the number. But if you take the deep dive and you look at this map, it's like, wow, they're not all 1 in 31. They're all really, there's a lot of variance. And that's probably influenced by early detection programs and funding for the state and various things that impact why the numbers are different. And here, if we take a little bit of a closer look, this is looking at the prevalence for each state. You can see California on the right with the highest prevalence and Texas, Laredo on the left with the lowest. But what I like about this, and what I like about the CDC's out-of-network report in the MMWR, is that there's a lot of transparency. Here you can see, because you might be thinking, oh, well, maybe, these children are being counted if they just have an ICD code in their record. But no, this graph, this bar graph on the left shows you that the majority of children, as designated by the green part of the bars, that shows you the percentage of kids in that state that actually had an ASD diagnostic statement based on a professional evaluation. And you can see that there's there's a lot of kids that actually had their record counted based on that. Then the blue represents ASD special education eligibility, and then only a tiny portion, the purple, are kids that were counted based on an ICD code only, maybe from a billing record or some other administrative record. And here, again, the reason I think San Diego is high is because we're very fortunate that we have a Department of Developmental Services, and we have, I think, over 20 regional centers across the state whose mission is to serve children with autism, to help provide diagnostic evaluations and interventions and coordinate services for kids. And so we have a really active system to help serve families that have a child with autism. So we've got a lot of cases here that are being served. And I started a Get Set Early network with 232 pediatricians that also screen for autism at 12, 18, and 24 months. And then they come to our center. And so we just have a lot going on here in California to serve children. And if you look at this Euler plot, this kind of gives you a little bit more insight into this idea about the types of records that children have when they are counted as an ASD case. So 68% of kids had a diagnostic statement in their files, and 70% of children had two out of the three types of identification. So that means that there's replicability. So maybe they had a psychological report saying autism, and then they also had an autism eligibility in their school. So that's nice that, about 2/3 of the kids are having ASD mentioned in multiple places in multiple agencies across the geographic region. 34.6% of kids actually had ASD recognized with all three types of ASD identification, and only 9% had an ICD code only. So this kind of just gives you an idea of sort of the rigor of which a child is considered to be counted as ASD. And for those of you who are kind of nerdy and like all the details, Table 1 in the main paper, it lists each of the states, and then it tells you things like the surveillance region for each state, the type of data sources used. Mostly everybody uses health, which is like a hospital record, education, which is schools, and then sometimes also like treatment agency records. This tells you the total population, which in each of the states, the number of cases of autism that were identified, and then finally the prevalence for each state. So you can check out Table 1. But obviously, we are here today, probably most of us, because the prevalence has been increasing a lot across the past 22 years since the CDC has been counting autism and determining prevalence. It started out in 2000 when the CDC first started as 1 in 150, and it keeps going up and up and up. And finally, we get to today where we are at 1 in 31. So what's going on? Before we answer that question, not that you can fully answer it, but before we talk about it, I thought we should really think about global prevalence rates, because if we're the only ones with this really high rate, then maybe there could be something specific to America in terms of some environmental issue going on. But if it's also very similar across the world, then maybe we need to broaden our thinking about what's going on. And so here we have a map, and this is just a quick snapshot of some papers, but basically, if you look across the globe, all these are different prevalence numbers, and there's just like across America, there's lots of heterogeneity. But I think what's important to take away from this is there are some places that are definitely have, much higher prevalence, like, the rates are 1 in 333 versus, so lower prevalence, versus Iceland, which is about the same as us, Nigeria, similar to us, and some places that even have higher prevalence, like Australia, it's 1 in 23. So this is not a US phenomenon, this is a global phenomenon. And so when you think about, okay, what could be contributing to this? There are at least 11, way more once we start getting into the genetics and biology, but just sort of some top level contributors. Obviously, there's improved awareness. And it's not just that, obviously pediatricians are now more well-trained and everybody knows about autism. It's improved awareness and reduced stigma. And I can't underestimate to you, how important this actually is. Autism is everywhere. Here is a little clip of Bill Gates from Microsoft talking about the fact that he thinks he has autism.
That if you were a kid today, you would probably have a diagnosis of autism. What makes you think that?
Well, the term, particularly when they say on the spectrum, that's gone from being a very narrow thing to now pretty broad. And I was very slow to develop socially. I kind of rock. I concentrate intensely. My teeth.
And now we have Elon Musk also talking about it.
I'm actually making history tonight as the first person with Asperger's to host SNL.
So autism is everywhere. We actually, if you haven't seen Love on the Spectrum, it's a fantastic show. I highly recommend watching it. And autism, we have this great neurodiversity movement now. Everybody is really talking about, let's make sure that we are inclusive and that we really respect everyone's differences. If you go on Amazon, when I first started, there were probably a dozen books on autism. Now, if you type in autism, there are 60,000 hits. Right? Autism is ubiquitous in our culture, in our media, in everything. And so we also have an expanded DSM definition. So if we think about the history of autism, you have to start with Leo Kanner in 1943, where he had his seminal paper called Autistic Disturbances of Affective Contact. And he really described individuals who were really intensely impacted. So here's his description. You know, it's actually a case study. He describes like 11 children with autism, 8 males, 3 females, and describes like children, you know, this particular child was happiest when left alone, drawing into a shell and living within themselves, oblivious to everything around them, right? So that's a pretty severe description. It took almost 40 years before autism actually got into the DSM. It first appears in the DSM-3 and then in the DSM-3 revised edition. But now we have what's called pervasive developmental disorders. And essentially, this was during a time period where autism was still considered very rare. There are some studies suggesting it was 3 in 10,000 or 7 in 10,000, right? So this is not something people really knew about. And the DSM-3 revised edition basically just had two categories under pervasive developmental disorders: infantile autism and PDD-NOS. Then we move along to DSM-4. We still have this category of pervasive developmental disorders, but now we have, 5 categories, and we have onset prior to 3 years, and you have to have an impairment in communication in order to get a diagnosis of autism. So now fast forward to the DSM-5, completely changed. Now it's just autism spectrum disorder. You don't have to have onset prior to 3 years. It can be currently or even just by history. Maybe you feel you're not showing symptoms now, but if you once did, you still can get a diagnosis of autism. There's no requirement for impairments in communication in the DSM-5. And the DSM-5 states that individuals with a DSM-4 diagnosis of Asperger's, PD-NOS, should all just be given a diagnosis of autism spectrum disorder. And so what's unique and important, I think, to understand about this is, yes, there's a broadening definition, but also the DSM-5 for the first time introduces this concept of levels. And it's really basically about how much sort of help the individual needs or support, and level 3 being the most severe. The other thing that's really important is the DSM-3 and the DSM-4, they're considered sort of categorical definitions or categorical ways to understand autism, where it's just one in a bunch of categories. Whereas the DSM-5 is completely different. It's dimensional. You have the spectrum, and then everybody is in this spectrum. And I think that this is where some of the debate and issues are coming in, this idea that autism is dimensional, and you have somebody who's going to go to college or get a PhD having the same diagnosis as somebody who's completely nonverbal, can't talk, and needs care 24 hours a day. So if I'm not going to go through all the different ones, but briefly, we have the improved awareness, reduced stigma, DSM-5 expanded definition. Premature babies are now living longer. We've got greater medical care, and there's an increased probability that you may end up with autism if you're premature. Like I said, we have lots of programs. We have universal screening programs like my Get Set Early program. That's contributing to some increased awareness. We have older parents. There's a link between maternal age and risk for autism. Also, children that were previously considered just having a global developmental delay or some sort of developmental delay back in the day when people were looking at records are now recognized as autism. So it's kind of a diagnostic clarity that's happening. There's also a huge surge in autism not-for-profits and advocacy groups. The biggest thing for not every state, but in California, insurance now covers autism treatment. So if you're a mom and you have a child who's showing some issues, maybe before you wouldn't run out and get a diagnosis, but if you know you're going to be able to get support for intervention for your child, you're going to get that diagnosis and you're going to go and get your intervention most likely. And that has led to an explosion of autism treatment agencies. And then one of the biggest reasons that nobody talks about, but I'm going to mention it because I think it's a huge reason, is the digital revolution. Back when the CDC first started counting autism, it was based on paper records, literally going and requesting records and looking through saying, oh, is there a diagnostic statement for autism? And here's a CDC data abstractor from the year 2000. Here's a CDC data abstractor today. Okay, say no more. You get the point, right? It's a huge factor. We can just press a button and get the data. But there's still, yes, all of these 11 reasons that I put down are valid, totally true. But could there be something else going on? Maybe in the environment? What are we knowing about genetics? Let's dive deeper into this why question. Okay, before we, you have to have the starting point when you think about autism and what's going on is genetics. We know that the concordance rate for autism, if you have monozygotic twins, which means that they have the same genetic, same DNA profile, is the concordance rate is 60 to 90%, meaning that if one child has autism, there's 60 to 90% chance that the other child will too. And there are lots of studies that have talked about this and reported this, but it's not 100%. And in fact, some studies say it's as low as 60%. So that means that environment or something else is playing a role here because it's not 100%. And that's important to kind of remember. And so we have to start thinking about genetics. And this is where Spark comes in. Spark has done an enormous public service, really extremely unbelievable, the number of individuals that they have gotten to join the Spark consortium and to provide us saliva samples so we can really understand the genetics of autism. And this is really a landmark paper that recently came out by Jessica Wright and everybody at the Spark team and Wendy Chung. And what they did was they did microarray genotyping and exome sequencing in 21,532 individuals with autism. And what they found is they found genetic differences in about 8.6% of individuals with autism. So that means like around 90% of individuals with autism currently don't necessarily have a genetic explanation for their autism. That's a huge chunk of individuals. So I look at this, and this is again my interpretation, is that autism is probably not at this point a single gene disorder. And to help people understand what's going on in autism, Spark put out this really lovely flyer called Understanding Spark and Genetics. And they have this nice illustration where we have these 3 glasses of water, and then you have this dashed line, which is a diagnosis for autism threshold. And in the first glass of water, if you have a de novo variant, which is essentially a genetic change that happens in the mother's egg or the dad's sperm, it's a de novo issue that happens. It is not passed down from parents. That's happening maybe in around 8-ish% of individuals, right? And that's likely the minority of individuals have this kind of single gene cause for their autism. Then you've got these rare moderate effects that happen sometimes, but it's pretty rare. And so likely the majority of individuals with autism, their autism is because of probably common variants, their background genetics that they inherited from mom and dad, that either alone, if you get that right combination, you end up with autism, or if you get that right combination with some sort of environmental thing that is happening alongside that, that's where you end up with autism. And it could be maternal health issues like a virus exposure, obesity, diabetes, infection during pregnancy for the mother. Environmental toxins has really been big in the conversation right now. Pesticides, heavy metals, BPA, microplastics, air pollution. Or it could be some sort of a medically related issue, like there's been an increase in premature births, and then we're also much better at keeping premature babies alive and healthy. That could be a contributor. We also have a big surge in IVF and fertility assisted pregnancies. Like I said, we have parents are waiting until they're a lot older to have children, and so advanced maternal paternal age could also be a factor. And so at our center, we recognize that autism is strongly genetic, highly heritable. But in order to start figuring out what's going on, we really need to understand the basic biology. So then once we understand the biological profile, then maybe we can start figuring out what gene pathways, what environmental teratogens to kind of look for. So our center has really been focused on understanding how the brain develops. And in 2011, Eric Crochane and colleagues published a paper in JAMA, a landmark paper, basically where they had some postmortem tissue of individuals with autism who had passed away, sent it out to stereologists who were blind to diagnosis. We had a few autism in control cases, and they just basically counted the number of neurons in dorsolateral prefrontal cortex. And here on the y-axis, you can see the neuron count in billions. And you can see here that this is the mean line for, actually that's the median line for individuals with autism. And you can see here on the right, you have control cases where the overwhelming majority of control cases is lower than the average for autism, the median for autism. And in fact, so we have less than 1 billion neurons in dorsolateral prefrontal cortex in normal controls, neurotypical controls, in contrast to over 1.5 billion in autism. So individuals with autism have a lot more brain cells. And we'll talk about, in some cases, it's probably great. It probably helps individuals, which is why we have, amazing individuals with autism. But sometimes if you have too many brain cells, you're going to have noisy connections and inefficient connections, and it's not always great. So we don't know for sure, but we think that that's kind of a clue. And if you and because brain cells are generated right during prenatal life, aside from maybe the hippocampus, you might be able to grow a few more, but for the most part, we are born with all the brain cells we will ever have. And so the fact that individuals with autism have more brain cells means something was happening during pregnancy. And most people now recognize that autism begins during prenatal life. And if you go into PubMed and type in autism prenatal, you're going to get 3,555 results or thereabouts. And so we kind of followed up this study. Actually, we've been using not only postmortem research, but also MRI. We published a study in 2001 called Unusual Brain Growth Patterns in Early Life in Patients with Autistic Disorder, where we looked at toddlers using MRI volumes and found a 12% increase in gray matter volume. And this has been replicated as reviewed by Sacco and colleagues by over 44 MRI studies. So it's definitely something that is replicable across the country. Again, this is the postmortem study we talked about. And now our center is really kind of diving into this idea of understanding how the brain develops using brain cortical organoids. And there's a paper recently published by Eric Crochane and colleagues, and also an earlier paper by Marchetto and Crochane and colleagues, showing that brain cortical organoids are about 39% larger in autism. And so here we are really trying to push for this idea of looking at the autism living biology by using an approach where we have toddlers, they come to our center and we get a blood sample. Here I did it the reverse because I thought it was cuter, but this baby who gets the blood samples, and you just basically select out the PBMCs, peripheral blood mononuclear cells. You can't just use any blood cell, you have to use PBMCs. And the PBMCs are converted to iPSCs, and then that's induced pluripotent cells, and then those generate neuroprogenitor cells, which turn into neurons, and then you can make brain cortical organoids. And, depending on the lab, people will grow these out for 10, 15, 16 weeks, and then you look at these brain cortical organoids. And basically, I want you to look at this work that Eric Crochane and Mel Campbell and Bonnie Taluja and Alice Motri are doing, and me, but really them. I am the support team when it comes to the basic biology here. Right here, this is basically what you're seeing on your screen is DIV means days in vitro. And when you look at the brain cortical organoids of a single ASD toddler, this is days in vitro 3, 6, 9, 12, so on and so forth. This is the size of the brain cortical organoid 24 days after being in that petri dish. And this is a typical brain cortical organoid after 24 days. What do you see? Right? You can just kind of see by that it's magnified, but clearly the BCO is larger in the ASD individual. And if we look at some other studies to kind of put this into perspective, this is a study by Marchetto and Crochane and Motri and lots of people where this is just a 2D image. So this is not a 3D brain cortical organoid. This is just a 2D image looking at cells. And you can see that there's a higher density of cells in the ASD individual relative to control. And when you do a correlation with MRI, basically brain volume, we were lucky that we also had MRI scans on these children. You can see that there's a correlation between brain volume and number of cells in children with autism. And here's our more recent study by Crochane et al. In 2024, molecular autism. And again, you can see enlarged BCO volume, but then we also are looking for relationships with phenotype because we want to understand, I think if you're trying to understand the causes of autism and what the drivers are, I think looking for relationships with social attention and social engagement, which are areas which are foundationally challenged in individuals with autism. And here you have a really strong correlation. And these are just basically control individuals. Neurotypical toddlers are in green. Mild ASD cases are in pink, and individuals who have profound autism based on their cognitive profiles are in purple. And you can see this relationship where the bigger BCO size, the more social challenges you have. So that's why we're really thinking this is pretty foundational to what's going on in autism. And so the MMWR paper also talks about age of first diagnosis, which is very critical to me because those of you watching who know me, I am a big time early identification researcher using I developed the Get Set Early model to work with pediatricians to mobilize them to try to find autism as young as possible. And we're catching kids as young as 12 months. But if you just look across, the CDC report has a median age of first diagnosis changed. You can see here on the x-axis, it goes from 2000 all the way to 2022. Hasn't changed a huge amount. It's going down a little bit, and partially that might be because California recently joined the network and we have a much younger age of first diagnosis. But overall, it's still around age 4. And again, the reason we have these lower ages is because of the San Diego Regional Center. Here, our median age of first diagnosis is 36 months, but kids who come to our center are much younger. We are doing an average of 17 months, some kids coming as young as 12 months, and then we get the kids into immediate treatment just to help facilitate their language and their social engagement skills so they can hopefully take off and just really achieve all the goals that they want. And again, this is because of the Get Set Early Pediatrician Network. And again, there's always variance across the network. In this graph, you can see the median age of first diagnosis across all the sites, with Texas Laredo having the oldest age at almost 70 months. Everybody else is kind of in the 40s. The reason this is so important, I know everybody says this, but I'm going to really try to show you some slides that really bring this point home, is that brain plasticity is much higher at younger ages, right? Think about trying to learn a language at age 20 versus age 2. You do it without even thinking. At 20, it's a task. It's a hard job. And that's because, the brain is much more responsive to change, and you can establish new connections much more easily and prune away ones that aren't working much more easily when you're younger than when you're older. So to kind of drive this point home, I've shown different slides in different lectures, and I switched it up this time. This is actually a stained image from frontal cortex from layers 3 through 5. We do have 6 layers of cortex, but this is just a zoom in because I thought this was a beautiful image showing. I mean, it doesn't have the resolution. You can't really quite see the synapses, which are the connections between brain cells, but you can see this dense architecture and how the cells communicate. And here is a landmark paper by Huttenlocher in 1997 where it's actually showing this. We have a huge burst in synaptogenesis, right, that happens during the first years of life. Here you can see conceptual age is on the x-axis, and newborn period is here shown in the red line. So this is conceptual age. So it's through, I think around babies are born about conceptual age 280. So that's why the birth line is there. And then he's looking at the number of synapses in different brain regions across different ages. So we have visual cortex is shown in the white open circle, auditory cortex is in the black, and frontal cortex is shown with the X's. And you can see these different peaks. Auditory cortex peaks in the number of synapses, around 3 months. Visual peaks by around 1 year, and frontal lobes peaks around 3 years. So there's a huge amount of activities that's going on. And if you think about it, when you make a new memory, when you learn a fact, when you learn anything, it is because synapses, through cell assemblies, lots of synapses are strengthened, and then you learn something again, you see it again, and that whole the synapses in that pathway are strengthened, and they keep being strengthened the more you learn something. And it's really a profound thing that is happening during early development, which becomes less with age. And so that's why, I think having early intervention, it doesn't have to be, you don't have to use the label autism, just to kind of get the kid engaged so that they will be able to optimize their skills to go off and do whatever they want in life. And this is the time to do it super early. And so there are a couple of papers that really show this out. Like, for example, there's a paper in JAMA Pediatrics by Whitehouse and colleagues that shows that early preemptive interventions starting between 9 and 12 months is associated with reduced symptom severity, reduced odds of an ASD diagnosis at 3 years in children at high risk for autism by virtue of having an older sibling with autism. There's also another paper that came out by Guthrie, Weatherby, and Lord's group recently in 2023, where they showed, it was a randomized control trial design, where they showed that children that started the treatment at either 18 or 27 months of age, and the children who started at 18 months actually had much stronger gains in receptive language than the children who just started it like a year later. Both kids, both groups did make improvements, but it was a larger improvement in the earlier group. And what's also, I think, important to think about is just kind of go beyond autism and what do we know about the impact of early experience and engagement in other fields. I really love talking about this paper by Sejas and colleagues in 2023, where basically we have some babies that are born with hearing loss. And some babies will get their cochlear implants pretty immediately, and others, it might happen later. And then we can test their scores. So basically, this is basically an intervention that is rescuing hearing so that you can hear the human voice. And we have children who had their hearing corrected prior to 18 months are shown here in the orange circle. And then children who got their hearing corrected later or not at all are shown here in these other triangles. And what you can kind of see is across all these different domains, children who got the intervention, right, which has had their hearing restored prior to 18 months are doing way better in language, math, reading, writing, and science than children who didn't get that. So there's this kind of critical period during very early development for many things, including language development and social development. There are also children who are born with cataracts. If they get them removed later, they have really issues with face perception versus children who got the cataract removed immediately. That's a paper published by Legrand in Nature. Another, landmark paper is by Chuck Nelson's group. You may have heard about this, where there are individual children who were placed in an orphanage at a very young age, mostly starting from birth. And some children got out of the orphanage at earlier ages. Some had to stay in there. And this orphanage really was sort of, I think, experientially, it was a deprived environment, right? You can see there's no pictures on the walls, not a lot of care, not a lot of experiences that are enriching for the children. And what this showed is that children who got out of that orphanage and into foster care super young had a DQ, developmental quotient, of 94.9. So let's say 95, versus children who stayed in and didn't leave until they were 2.5. That's 15 points difference for IQ, which is huge. Again, underscoring the so important of this early period of life. And so, because this is near and dear to my heart, I actually started my career in the field of treatment. I was doing all sorts of treatments here at UCSD with Laura Schreibman. I eventually decided I wanted to understand neuroscience in the brain, so I kind of switched. But I've always sort of had this intersection and this really love of early intervention. And before you can, provide intervention to a child, you have to find them, right? And diagnoses are expensive. There's long waiting lists. So I've been really working a lot on eye tracking technology as a tool to potentially identify autism at the earliest ages possible and then get kids out into intervention. Here, if you don't know how eye tracking works, a baby sits down on the mother's lap. They're just kind of watching a movie. There's an infrared light beam that is emitted from the eye tracking machine that creates a corneal reflection on the baby's eyes. And then that light is read back into the machine. And basically, there's also cameras that take pictures of how the eyes are moving. And then the gaze point is calculated based on the vector between the pupil center and that corneal reflection. I don't know if you can see it, but it's like this little thing right here is a corneal reflection. And that vector tells you where the baby is looking. So we've been doing a lot of work on this, and we developed what's called the geometric preference test for autism. It's literally just a 1-minute eye tracking test where a child they just sit there and they're watching a movie, and they can look at the little kids or they can look at the shapes. It's visual only, there's no sound, and it also kind of takes advantage of the fact that some kids with autism are attracted to kind of repetitive movements, and they have enhanced visuospatial abilities and interest in repetition, maybe superior math. So we have the social area of interest and the non-social area of interest, and we just basically calculate the amount of time a child's looking at one area versus another area. And here is some data from a neurotypical toddler at 14 months. The red dot actually is the data. So the baby is watching this movie. They don't see the red dot. You're seeing the red dot because that is the neurotypical baby's point of gaze. And it's you can see across this 1 minute, the baby kind of looks around, but he's spending more of his time looking at the little kids moving around. Then we have a toddler with autism at 15 months, and see if you can see the difference in his gaze patterns. Right, so you can probably tell the difference. So this is the GeoPrep test. It's our first eye tracking test. We had a paper with 1,863 toddlers. This is the raw data results. I want to make sure I have time to go over lots of other stuff, but basically, if you just kind of look at this magic line, and all here on the x-axis, you have all the different types of children that participated, like children with autism and TD children, right? Who's up here looking at geometric shapes more than 69% of the time? Is basically 125 children with autism did this, but in contrast to the 1,138 children that did not have autism in the study, only 29 children looked at the geometric shapes more than this threshold, which translates into specificity of 98%, sensitivity of 17% though. So that basically means it's got a good true negative rate, not a lot of errors, but it's only detecting around 17% of the population. We've replicated this, other people have replicated this, so it's a really robust finding. And I was super excited about the 98% specificity, but I wanted to increase the sensitivity. So what we're working on now is a Get Set Early multi-domain eye tracking test battery, where we've got 6 eye tracking tests, and it takes around 12 minutes to do them all. And we've got these 6, and basically 3 of them are dedicated to kind of visual social attention, because we wanted to develop eye tracking tests that tap into key areas of challenge for kids with autism. Some kids maybe don't make great eye contact, so they kind of more visual social attention issues. Other kids maybe have gaze shifting, so maybe their eye contact is good and they engage, but if you point or if there's like a busy conversation between people, they have a hard time shifting. So that's another social attention subtype that eye tracking is trying to quantify. And then we have kids that maybe are fine in those domains, but they're not listening to speech so much. If you call their name, they may not respond. They have a hard time processing language. So that's auditory social attention that we're tapping into using eye tracking. And we do it using gaze contingent technology, which basically means that when a baby looks at a particular area of the screen, they hear somebody reading them a story, versus if they look somewhere else, they'll hear like a car is going down a highway. So that's how we're measuring these things. And we combine all that to generate an autism score. So that tells the parents and the pediatricians scale of 50 to 100 is you're in the autism range, and 0 to 49.999 is you're not. So it's kind of clinically useful. We're working on it right now. So we got that sensitivity up from 17% with 1 test to around 60% with 6 tests. Still high specificity at around 92%, and positive predictive value still very high at around 90%. So we're super excited. Stay tuned for this. We're hoping to get this paper out very soon. And now let's talk about demographic trends. The CDC report talks and tells us a lot about sex differences. So back, let's say a decade ago, it was sex, ratios were 4.5 to 1. So around every 4.5 males, there was 1 female. Now it's going down. It's only 3.4 to 1. And so first blush, you might be thinking, well, that's great. We're identifying girls more, and this is fantastic. And that very well may be true, but we have to think about it a little bit more deeply. If it's basically, if the rate of males, if we just keep finding more and more males, even though we're finding more females, if the rate of the male finding is going up faster, that is also important to consider. So it's not necessarily the case that we're doing a great job with females. So I think an easier way to think about it is if you just look at this table right here. These are fake numbers. I made it up just for illustration. But let's say in 2018, there are 40 boys with autism per 1,000, and there were 10 girls. So, right, you do the math, that's a 4 to 1 ratio. Super easy. Then in 2022, let's say you found 68 boys with autism and 20 girls with autism, right? They both increased. But the boys increased more. They increased by 28, and the girls only increased by 10. But when you do the numbers and calculate the ratio looks better. It's like 3.4 to 1. So, I raised this just because I think it's important to kind of think deeply about all these numbers when we're looking at them, although I am excited that the ratio is going down for sure. But if boys are consistently more identified than girls, and does this mean that girls are milder in their clinical presentation? So we have a paper published by Nazari and colleagues, and me and Eric and everybody in our lab, 2025, where we looked at the clinical scores of 2,618 toddlers, mean age 27 months. Most of these toddlers actually came in multiple times by licensed psychologists and have their diagnosis in our database. And we basically said, okay, are boys or girls showing more symptoms? And here on the left are scores for kids with autism, and we have them broken down by adaptive or language or motor or other or social. And here this dashed line represents a difference between males and females. If a bar does not go past this dashed line, that means there's no difference. And basically, out of all of these, there's like 18 things that we were looking at. In autism, only daily living skills were slightly better in females. Whereas if you look at the typical kids and you look what was significant, there were almost everything was better in females than males. So basically, the old sort of cliche that little girls develop faster is true in our data set. But as it relates to autism, clinically, they look similar. And you have to remember that we're using the Get Set Early model at our center, where it's a population-based screening approach. Parents are filling out a questionnaire, then they come to us. So it's not as biased as a fully clinic referred sample. So we're thinking that maybe there are differences later in life in autism, but they don't appear to be there at the time of first diagnosis. And that's important, I think, for the conversation. Then when we look at other demographic trends, the CDC paper tells us that for the first time, actually this has been happening slowly across other CDC reports, but basically in the past, children who were from African-American homes or Hispanic homes were actually under-identified. They were not getting counted, and they weren't getting their services. But now there's a reversal. For the first time, we have Asian and Pacific Islanders actually have the highest prevalence rates, whereas white kids have the lowest prevalence rates. Again, this is not the very first time. This trend's slowly been happening, but it's pretty clear here in this most recent report. And this is in contrast to the way things used to be in the past, where it was a complete opposite. We also see the opposite trends now in SES, where in the past, higher SES had the highest prevalence, but now they have the lowest prevalence. And so I think this is reflective of really all of the great work that everybody's been doing to try to ensure equity for everybody. And so the last part I want to talk about before we jump off into some questions is the CDC report where they talk about the clinical characterization of autism. This is just a nice little donut plot where we are looking at individuals who are tracked and what their cognitive and intellectual levels are. So you can see that the blue represents kids, individuals who have IQs at or below 70, and that's 40%. Then we have kids who have IQs between 71 and 85, that's orange, and kids who had IQs above 85 is 36% of the ASD population, and that is shown in gray. And again, here we have this. But if you combine the kids who are less than 70 and 71 to 85, that's 64% of individuals still probably need some help. And within this percentage of kids who have IQs below 70, there are kids who really are quite challenged. And so this is part of why individuals, there are some people who are saying, okay, let's unpack this a little bit more, because you have kids with IQs of 120 alongside kids with, who are very challenged, who have IQs below 50, and they have very different support needs. And so really it's only around a third of individuals on the spectrum have IQs at 85 or higher. So this moves into this idea about profound autism. And you probably know that in 2022, Kathy Lord and colleagues wrote this really elegant paper where they just basically threw it out there and said, okay, there's around, I think they were estimating, between 25 and 50% of kids with autism required 24-hour care. They have IQs below 50 and they are either minimally verbal or have no verbal skills at all. And that's how they defined profound autism. And it started this huge conversation. Hughes, just a year later, took the CDC data and said around 26% of kids with autism actually meet criteria for profound autism using those criteria. Then we have different autism societies coming out with statements. The Autism Science Foundation thinks that it's important to really use the label profound autism because it will provide critical specificity to the extremely broad spectrum. It will simplify the process of determining appropriate care, leading to quicker and more precise interventions. The Profound Autism Alliance says it's an administrative term, it's not a diagnosis, so they're kind of in the middle here in this conversation. And the National Council on Severe Autism, their position statement says that there is a need for categorical recognition of severe autism in the DSM. So they didn't necessarily come out and say it should be its own diagnostic category, maybe it's a specifier, but people are really pushing for this distinction because it will really help kids be identified immediately that fall into that category and get the intervention that they need. And it's such a big talking point that the New York Times actually published something just a few weeks ago. Should the autism spectrum be split apart? They highlighted Allison Singer, who's president of the Autism Science Foundation, and she's quoted as saying that the idea that Elon Musk has the same diagnosis as my daughter is ludicrous. Right, so this is just an example of why this is such a huge topic right now, and it's very important because people feel frustrated if you have a child who is profound and you're not, it takes forever to get the care that you need. But if you just had, some specifier, maybe that care would come more quickly. So is the DSM sex, which I think is coming up in 2026, what are they going to do about it? Right now, like I said, we have these 3 levels, with level 3 being the most severe, but that's vague. Like maybe people don't even remember which one's more severe. So maybe they'll do something like mild, moderate, profound specifiers. Maybe they'll have a completely new diagnosis. We will find out. And so this last slide before we have just a few minutes for questions after my last slide. This heterogeneity is so important. And as I said, social challenges are at the heart of autism. And I've been, like I said, developing eye tracking, not for just diagnosis, but for me, it's really more about precision medicine and understanding the heterogeneity. Because some kids with autism actually have pretty good social attention, and other kids, it's more severe. And those are the kids that really are going to need help. And here, this is a study where we just looked at the eye tracking of 173 kids with autism, 24 who are neurotypical, and 30 who had a non-ASD delay. And these are kids who took at least 5 of the eye tracking tests. And we asked the question, do they, how many kids are consistently showing low social attention, like across every test you give them, versus how many kids with autism are just showing really good social attention across like every test that you give them, right? We're trying to look for intra-child replication to get a handle on the heterogeneity in social attention, because really it's hard to do. Like you can't just give everybody 8 ounces all the time. It's time consuming, and eye tracking is great because it's quantitative. Every kid gets a number. This is your social attention score for this test and this test and this test. So here, we found here, this just shows the percent fixation on social images or social sounds or social points across different eye tracking tests. For this is just a random sample of 10 kids. And these are kids that I would say have low social attention. That is because you can see percent fixation on social fell below 50% on every test that they took, right? But this is just 11% of the ASD population has pretty profound social attention challenges. And here, if you look at these other kids, 23% of kids with autism actually have really good social attention, and they did spectacular on every single eye tracking test that we gave them. And the reason this is important to me is because I believe maybe there might be different interventions that you might want to use. Maybe these kids would perform better just with more academics and kind of more common sense types of interventions to help them connect with people socially, but not all this, kind of autism relevant social attention training, because they already have pretty decent social attention versus these kids that have a lot more challenges. But overall, the majority of kids with autism, 66% were mixed. So and these actually correlate with symptom severity. So basically, the kids who have more profound attention issues have the lowest expressive language, the lowest cognitive ability based on the early learning composite. And again, we're trying to find these eye tracking based subtypes to parse heterogeneity to move towards precision medicine. So with that, I know it was a lot. I had a great time putting this together and talking with you. And I want to thank you for listening, all the families who participate in our studies, all the pediatricians in our network, our funding agencies, Eric Crochet, the lab co-director, and everybody who works with us. So thank you so much.
Thank you so much, Dr. Pierce. I think this is definitely a lot of information to get through in an hour, and I think you did a really nice job summarizing a really hot topic right now, to say the least. So we have a couple minutes for questions, and I will, we'll get through probably a couple here. So I think one thing that came up from a few people, both leading up to the webinar and today while we were live, is what about adults? So we're looking at the CDC numbers, looking at 8-year-olds. How are, so if we're looking at 8-year-olds, is the actual rate in the US perhaps much higher than what's being reported by the CDC? And then sort of a follow-up question to that is, how are those who were diagnosed in adulthood factored into these prevalence statistics?
That is a fantastic question, and I didn't mention it, but the CDC ADDM network, there's a couple of sites that actually also track 16-year-olds. Now, they're not like adults, but that is emerging towards adulthood. And we do have prevalence estimates for that age, which are very similar. And so that is how the CDC is handling sort of the adult end of the spectrum. And if we get more funding, then it would be fantastic to do it in 20 or 30-year-olds. But as I mentioned at the beginning of the webinar, it's incredibly time consuming and meticulous because we have the geographic regions, and you get the relationships, and you get the data, and you do all the stuff. So, every age that you do is an enormous workforce associated with that. So the CDC tracks 4-year-olds, 8-year-olds, and 16-year-olds. The 16-year-old network is a little bit smaller because of expense, but we do track that as well. And the numbers are similar at that age, and we would love to go higher. But that's a really important point, because some people, like I showed in the video, like Bill Gates, maybe at 30 decide maybe I have autism. A lot of people, because of all this awareness, have an awakening, and they're getting their diagnoses much older and later. And I agree, we don't have that much research. We'd love to also track older ages more.
Yeah, and I think that's a great point that you bring up too, is just like this increased awareness, whether that's from parents to then take their children to, perhaps get a diagnosis, or just adults taking themselves. And so do you think that really is the biggest cause of the increased rate, or is it really predominantly awareness based, or is there something else, something bigger that you think maybe is, causing
Yeah,
harm?
I mean, I thought about it long and hard, and I made that slide. There were a lot of slides, but one of my slides was I thought about the top 11 reasons, and I believe in all of them. And I do, the reason I put awareness and reduce stigma first, because I think that that is a huge contributor. Like I said, you've got Elon Musk going on Saturday Night Live, being like, I'm, on the spectrum. It's everybody's talking about it. I have like a neighbor's like, I think I'm on the spectrum. It's because of all of it's part of our social fiber now. I mean, up to 5% of the population has autism. It's just a thing. It's just, part of the spectrum of being human. And I think a lot of that awareness has a huge amount to do with it. But like I said, I tried to dive deeper and think about there could be interactions with different environmental factors, pesticides or BPAs or plastics. I mean, there are even microplastics in breast milk, in placental tissue, unbelievably so. It's everywhere. And so, we can't discount that some of these changes in how humans operate, it could potentially be interacting with genetics to be, having some role to play in what's going on here. But I think, where all the 11 factors I mentioned, I think are really the pushers.
Great, thank you. And I think in this last minute, I want to, take advantage of the fact that we have you here, and you talked about the eye tracking that you've done, and we did get a lot of questions about, how people can participate in this, or some pediatricians asking how they can utilize the Get Set Early method. So can you just talk a little bit more briefly if that is something that is
Oh my gosh,
available
I'm so
outside of
excited.
California?
Okay, if you are a pediatrician, email me because we have an NIH grant right now to test the efficacy of eye tracking in, well, maybe checkups in 4 different pediatric offices throughout San Diego County. Would love to do more. It's obviously just very expensive to do it, the eye tracking system and everything like that. So, but if you'd like to do it, just email me, kpierce@health@ucsda.edu. The eye tracking technology is licensed through the university. It's also free for use for research. So anybody who wants to use it for free, only for research, you can email me. But if you are wanting to like use it commercially or something like that, it is licensed, and we do have a company in India, Butterfly Learnings, that has licensed the technology, and they're trying to see if we can have it be used as kind of, across different regions, hopefully worldwide at some point. But right now it's really in the research domain, and we're just starting to try to see if we can incorporate that into the public sphere.